Okay, welcome to AQA A Level Biology Paper 3 Essays. This is my second video dedicated to an essay. So there's all sorts of titles that have come up over the last 30 years, 15, 20 years, whatever. There's hundreds of these titles. And AQA actually make these titles public so you can find them online. The one we're going to look at today is write an essay on energy transfers which take place inside living organisms. So within living organisms, there's a very similar one which is between living organisms, but we're looking at ones in living organisms. Okay, so when you're going to write an essay on the importance of energy transfers within living organisms, you need to do your plan. And I would recommend spending 2 minutes because you might find out, damn, I don't know anything about this. So your goal is to write an awesome essay. You absolutely have to spend a couple of minutes brainstorming the topics. And then you want to carry out your action. You're going to spend 40 minutes writing the essay once you've done the plan. You always have time to abort. If you're on the plan, you can abort and switch to the other essay. Now, what does AO1 and AO2 mean? AO1 is scientific content. AO2 is basically the application of the content. And that is stressed in the word in the title, which is the importance. So the importance of energy transfers inside or within living organisms. I've changed the wording there a little bit, but that's because I'm an idiot. Anyway, so really, really important. If you're just brainstorming out the topics, like energy, when I think of the word energy, you should just be thinking ATP. And so if you see energy, just think of as many different topics you can think of where you can talk about ATP, because ATP is the energy carrying molecule in A-level biology and in biology. And so it's really the one that you've got tons to talk about: active transport, mass flow, selective reabsorption in the kidney, the loop of Henle, co-transport of glucose, formation of an action potential, muscle contraction, breaking those cross bridges in the actin and myosin binding sites, photosynthesis, the formation of ATP to deliver energy to GP to turn it into TP and produce organic compounds. So anywhere you can think of ATP, ATP is inside organisms, not outside. So we're talking about energy transfers in organisms, and ATP is the carrier molecule inside organisms. It's a different story if you get an essay on energy that is between the environment and the organisms, or in between organisms themselves. So in living organisms, adenosine triphosphate. Notice that once I've said the term, I can then use the abbreviation for the rest of my essay. So ATP is the main form in which energy is transferred. It's transferred in muscle contraction, nerve impulse transmission, photosynthesis, respiration, and during active transport. I'm just giving a waffly introduction that won't score marks. Okay, my first paragraph of 5. During the light-independent reaction of photosynthesis, the chlorophyll in photosystem 2 absorbs light in the red and blue wavelengths of the electromagnetic spectrum. This causes photoionization, a process where an electron from the chlorophyll is excited to a higher energy level. Some of this energy is used in photolysis, where water is broken into hydrogen ions, oxygen gas, and more electrons. The excited electrons from the chlorophyll then move down the electron transport chain via a series of redox reactions, I could have put that there, involving proteins embedded in the thylakoid membrane. That's the ETC. Okay, some of that energy is used for the active transport of hydrogen ions from the stroma into the thylakoid. This creates a concentration gradient for chemiosmosis. As the hydrogen ions diffuse through ATP synthase or ATPase, also embedded in the thylakoid membrane, the kinetic energy from the movement helps join ADP to PI to form ATP. So some of that original sunlight energy is now in the bonds of ATP. Some of the hydrogen ions and electrons go on to reduce NADP to NADP reduced. Then in the stroma, in the light-independent reaction, that original light energy that hit chlorophyll in photosystem 2 is being passed via ATP and NADP reduced to glycerate 3-phosphate, which is then reduced further to triose phosphate. So some of that energy is used to reduce the glycerate 3-phosphate and used to produce organic substances. You got your lipids in the waxy cuticle. You got amino acids by adding them to nitrates to make proteins that can be used for structure and growth and production of hormones like IAA in the plant and things like that. So your AO2 in this one is a little bit blended in with it. You can go off on your AO2, basically why it's important to produce these substances, why it's important for this energy transfer to take place. Notice that a lot of the content in the essays, as I go through about 30 of these videos, a lot of the content is going to be similar from video to video because you can apply the same knowledge in many different essays. So obviously I've got a little bit in purple here. This is supposed to be why kind of the energy transfer is important. If we didn't transfer energy to photolysis, we wouldn't resupply photosystem 2 with electrons, and therefore we would end up not being able to reduce any more NADP, and without reduced NADP we couldn't turn GP to TP, etc., and form those organic compounds like lipids and things like that. So this is just kind of giving a justification as to why the energy transfers are important, and you can just pick something which you can talk about that you haven't already talked about to do with the topic. So it's kind of like AO1 and AO2 there. Now, do you need to link the topics? Not really, but sometimes it helps your essay flow. So the bonds in the glucose produced by the plants can be considered to contain some original energy from the sunlight. This can be used in respiration by both plants and any animals that consume the plants. Now I'm going into respiration. So the glucose consumed by animals enters the cytoplasm of the animal cells. The glucose is phosphorylated by 2 molecules of ATP, which essentially transfer energy to produce a more reactive molecule. That's hexose bisphosphate. It's just a 6-carbon reactive molecule that quickly breaks down into pyruvate, releasing 4 molecules of ATP. So you end up with a net gain of 2 ATP. Also, 2 molecules of reduced NAD are formed, not NADP, that's in photosynthesis. And these 2 reduced NADs, they're coenzymes, and they can be used in anaerobic respiration to turn pyruvate to lactate and reform NAD, allowing glycolysis to continue. That's why that's important. The pyruvate can also enter the matrix of the mitochondria, be decarboxylated to acetyl-CoA in the link reaction. Remember, this is all the same energy that was released from the bonds and electrons of the glucose when we split it into pyruvate molecules. And it's then that energy is transferred to the bonds of the reduced NAD and the FAD and the electrons that have gone to them to reduce them, and hydrogen ions. So this energy is then transferred by those coenzymes to the electron transport chain. And the electrons from the coenzymes move down the proteins in the electron transport chain, which is in the inner membrane of the mitochondria, and they release that energy, which allows hydrogen ions into the intermembrane space of the cristae, which in turn leads to chemiosmosis of hydrogen ions through the ATP synthase, joining ADP to PA and forming lots of ATP. So in a way, I've kind of coloured in those purple because in a way that's my AO2 anyway. That's like my justification. All of that, I'm explaining why that transfer of energy in the Krebs cycle that came from pyruvate, why that transfer of energy was really important. So that's kind of like my AO2, because without it, we couldn't do chemiosmosis and make all those ATP molecules. So I'm then linking over to another paragraph where I'm going to talk about co-transport. So ATP is used in co-transport of glucose into epithelial cells of the ileum. First, the energy from ATP molecules is released when it is hydrolyzed to ADP plus PI, which powers the potassium-sodium or sodium-potassium protein pump. This pump pumps sodium ions into the blood vessels next to the epithelial cell, which helps creating a concentration gradient between the sodium ions in the lumen of the ileum and the epithelial cells, because the sodium's gone into the blood. So the sodium then diffuses in through a co-transporter protein down a gradient, and it brings the glucose along with it too. And then once the glucose is in the epithelial cell, it's in a high enough concentration to go by facilitated diffusion into the blood through a different channel protein. And why is this important? If we didn't have this process of taking glucose in against its concentration gradient from the lumen in co-transport, then we wouldn't absorb enough glucose. And we can sort of talk about net production of biomass here. Remember that net production is the ingested food by a consumer minus the feces and respiration. If we're losing a lot of glucose in the feces, we're going to lose loads of energy in the feces. We're going to have a lower net production and less biomass added to the consumer. So there's many processes like this one above. Within an organism that require energy transfer from ATP. So we could go on blah, 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 blah. And another topic, I don't know if this is my fourth or fifth, but is in the loop of Henle in the ascending limb. Sodium ions are pumped out into the medulla using active transport. This uses energy transferred from the hydrolysis of ATP to ADP plus PI. And the ascending limb is impermeable to water, so the solute concentration of the medulla is increasing as those ions are being pumped out. And this is lowering the water potential. This means that the collecting duct and distal convoluted tubule between them and the medulla is a steep water potential gradient. Should the osmoreceptors in the hypothalamus detect a low water potential, then ADH is released from the posterior pituitary gland, making the collecting duct and DCT more permeable to water, and this means more water is reabsorbed by osmosis into the medulla than into the blood. So this energy transfer that we talked about, ATP to ADP plus PI, was really important because it allows more water to be reabsorbed into the blood. So you're less likely to be dehydrated, which means you have more water available for hydrolysis reactions, catalyzing glycogen to glucose in glycogenolysis, breaking down proteins or polypeptides in the stomach into amino acids so that we can then use them in translation to make our own proteins. So that's all super, super important. Okay, so my fifth topic, I believe, is here. Implants the energy transfer of ATP to ADP plus PI. Notice I'm doing the same energy transfer here, but it's always for a different topic. So it is needed for active transport, again, in the active loading of sucrose into the sieve tube of the phloem. That lowers the water potential at the top of the sieve tube or at the source, and it moves water in from the xylem by osmosis down a water potential gradient. This is important, this energy transfer, because this ends up with a high hydrostatic pressure at the top of the phloem, at the top of the sieve tube, and the sucrose and amino acids then are forced down the phloem down a pressure gradient. This mechanism is known as mass transport. Okay, yeah. So without this mass transport flow mechanism, plants wouldn't be able to move their assimilates really long distances. And as they're multicellular organisms, they can't rely on simple diffusion alone to meet the metabolic demand. So this is all explaining why it's important that we have these energy transfers and that they play an important role in many, many physiological processes inside living organisms. Now we're going to beyond the specification. So beyond the specification, NAD, which we use in aerobic and anaerobic respiration to transfer energy, when we reduce it by adding electrons and hydrogen ions to reduce NAD, it's an energy carrier. It's made from vitamin B3, beyond the spec, known as nicotinamide, beyond the spec. Now, there's a disease called pellagra, which is a deficiency of vitamin B3 in the diet. And that's a little bit of a case study, this little bit of extra knowledge here, specifically to energy transfers. So, humans synthesize nicotinamide from an amino acid called tryptophan, and then the nicotinamide is used to make NAD. However, corn, which is the staple diet for a lot of people in the world, it's the staple diet of the Mexican Indians. It doesn't contain the amino acid tryptophan, which means they wouldn't be able to obtain NAD and make NAD reduced. Now, because they soak the corn in lime water and calcium hydroxide when being made, this adds tryptophan to the food and avoids them getting pellagra. However, in the United States, in the southern United States, the people didn't use lime water when making their tortilla chips. And this led to pellagra being endemic in the population. And what does that mean? That means that they weren't getting any nicotinamide B3, vitamin B3 in their diet, and they couldn't synthesize NAD, and therefore they couldn't make NAD reduced, and therefore the energy transfers for their processes would have been extremely badly affected, would have been far less efficient because they don't make enough NAD reduced, because they don't have the reactants to make NAD, which is nicotinamide. And this led to a large number of the population suffering from pellagra. The symptoms are diarrhea, dermatitis, and dementia. And so this just shows, again, it's a beyond the spec why energy transfers are really important. Sorry if this video is a bit rushed, but it could potentially come up in tomorrow's essay for the Biology Paper 3 for 2024. And they haven't asked about energy transfers in a long while. Just be aware, if they ask about energy transfers between the environment and an organism, as well as inside the organism, you can use a couple of examples of topics I've covered. There are lots of other topics you could use as well. But you can also talk about primary productivity and gross primary productivity with plants. You can talk about the nitrogen cycle if you're involving the environment. If you're doing between organisms, you can talk about saprobionts and how they decay matter in the nitrogen cycle. You can talk about pyramids of biomass and energy lost at each trophic level, and intensive farming and how important that is to reduce energy losses so that we pass on more energy to up the levels in the trophic levels, and why trophic levels are limited. You can talk about all the energy flow in the ecosystem as well. So you'd need to look at those topics as well, really, just to make sure that you're covered for energy transfers between organisms and between the environment and organisms. The ones in today's essay is strictly really talking about the transfers via ATP in the physiological processes within organisms. And of course, this essay would be perfect if they ask you an essay title about ATP. Look at all those examples of how ATP is useful that we've covered in this essay. So you can often apply many of the topics to many of the essays. All right, guys, good luck. Let's get to the marks.
You're still here? It's over.